The work that I do is related in large part to thalamic stimulation for epilepsy, and it covers scales from single neuron activity to local field potentials to network activity, and we'll go through all those things in turn. Um, so I don't have any financial interest related to this, but it is worth from a clinical aspect talking about the fact that most of the clinical applications that I'm talking to are cutting edge in the sense that many of them are not approved by the FDA, though some of them are under review by the FDA. So we'll talk a little bit about, what is brain stimulation for epilepsy. We'll talk a little bit about mechanisms, talk about some signal processing concepts, particularly in terms of connectivity. And then we'll talk about some physiologic cycles and how do we describe those in a quantitative way. So let's here's I'll start with the clinical background here. Epilepsy is a condition of seizures of repeated unprovoked seizures that have many causes, and this is a very heterogeneous disease, but it's also a very common disease. 1 in 26 people will develop epilepsy in their lifetime. Most people are well controlled on medications, but a third of people are not well controlled on medications and continue to have seizures despite optimal trials of medications. And epilepsy surgery has been studied for a long time. Wilder Penfield was one of the first, sort of big figures in epilepsy surgery. And so we've been doing surgery for epilepsy for, depending on how you count it, almost 100 years or more than 100 years. And that's mostly consisted of finding the area where the seizures come from and cutting it out. But more and more that becomes not a great solution, particularly because a lot of people have more complex seizure networks and multiple areas that give rise to seizures. And so, historically, if people went down this route of considering epilepsy surgery, 1 in 3 of them would be told that you're not a surgical candidate. Uh, and even amongst people with epilepsy, a third of them don't have focal epilepsy, meaning it doesn't come from a specific brain area. They have generalized epilepsy, meaning that they have some sort of typically what we think of as a genetic abnormality that predisposes all the neurons in their brain to hyperexcitability and seizures. And so this creates a need for other surgical treatments, which we have developed over many years, though many of these are much more recent developments. Um, when people come to see me in clinic and talk about why do surgery for epilepsy, because I think a lot of people intuitively understand that brain surgery is a scary and risky thing, is that seizures are also not good for you. So seizures are bad for cognition, they're bad for memory, and they're bad for quality of life, and they pose a real risk of death on a annual basis, particularly when seizures are uncontrolled. Okay, so three brain stimulation devices that we have for epilepsy. One of them is not direct brain stimulation, it's vagus nerve stimulation. Efferent transmission of signals from the vagus nerve to the brain. And then the things I'll talk about more are deep brain stimulation and responsive neurostimulation. Deep brain stimulation is designed for tremor and Parkinson's disease, and so it's stimulation of deep brain nuclei, including the thalamus, which is most of what I'm going to talk about by disruption of brain circuits, well understood brain circuits, whereas responsive neurostimulation is sort of what I think of as like a defibrillator, where you are recording brain activity, and then when you record a seizure or seizure-like activity, it delivers electrical activity to try to shock the brain out of this rhythm. We'll talk a little bit about mechanisms and why all of that is hocus pocus and what we can really understood to make things work better. Outcomes of stimulation is sort of a spectrum from less risky, less beneficial to more risky, more beneficial. So, vagus nerve stimulation, it's not brain surgery, but it also is the lowest response rate. Typically, we talk about response rates as percent reduction in seizure frequency. So, about half of people who get vagus nerve stimulation will have a half or more reduction in their seizure frequency. Deep brain stimulation, responsive neurostimulation. Depending on what time point you're looking at, you're usually talking about 70 to 80% reduction in seizure frequency, which is good, but it's not seizure freedom. It's not driving. It's not being able to reduce seizure medications in many cases. And then there's a significant portion of people who are non-responders and they go through all of this and they don't have a real clinical benefit. And then the last part of this too is that effects of stimulation don't happen right away. It often takes months to even years for people to see the material benefits of the surgical intervention that they have for epilepsy. So because all this is new technology, because our outcomes are good but not great, there's a lot of low-hanging fruit here, and there are a lot of assumptions that people have made about how these things work that are not true and potential sources for improvement in the technology. Okay, so I promised to talk about mechanisms. So deep brain stimulation, again, developed for movement disorders to try to interrupt pathological activity, like pathological activity that leads to tremor or rigidity in Parkinson's disease. And so the idea that was developed in Parkinson's disease was this idea of a circuit block that you were delivering high frequency stimulation to block transmission through a particular part of the brain. And this was taken from the idea that brain stimulation seems to do the same thing as creating a lesion in part of the brain, like in the thalamus or in the globus pallidus, one of these areas that's involved in the basal ganglia networks. And so, more than 20 years ago, some people thought that maybe we could do the same thing with epilepsy in well-described circuits. So, there's this Pepez circuit, which is a circuit that is really a memory circuit that involves the hippocampus. And the hippocampus is a common source of seizures and many forms of adult epilepsy. And so the question was, okay, so a seizure starts in this area, and maybe it doesn't become a problem until it spreads to other areas of the brain. So can we block the seizure before it spreads to enough parts of the brain for it to become a problem? So for it to become symptomatic, meaning that people notice symptoms from it, or generalized, meaning that it turns into a generalized convulsion. Now, if you did that, you figure you could just turn this thing on day one and that people wouldn't have symptomatic seizures anymore. But that's, again, not true. People don't have benefit from deep brain stimulation for epilepsy often for 6 months or more. So that is evidence against that idea. And then responsive neurostimulation, right? So this is a graphic, this is a schematic of brain activity, and then you have some sort of detected abnormal, for here, for example, low frequency, high amplitude oscillation, and you detect this activity, you deliver stimulation, and then the brain activity returns to normal. That's the defibrillator model that's how cardiac defibrillators work. But again, it's not consistent with what we see clinically. One, we rarely see this when we look at recordings from the device. Two, people might tell you that they have a seizure every day or a few seizures a week or seizures a month, but you see detections of seizure activity like hundreds of times a day, and not in people who are having hundreds of times a day. So that doesn't make sense either. And then again, you'd expect this to work immediately, but again, it takes months to even sometimes years for this to benefit. So let's talk a little bit about functional connectivity. Let's talk about this at like a 1990s level functional connectivity. So I'm going to talk about different nuclei of the thalamus here. The centromedian nucleus is the nucleus that's sort of crudely described on the left. This is actually from some of the work done by Hunter and Jasper in the late 1940s in cats. But looking at low frequency stimulation of the medial and intralaminar thalamus and what are the effects of that? So when Hunter and Jasper did this in the 40s, they were able to demonstrate that low frequency stimulation of the thalamus was actually able to evoke this very stereotyped 3 hertz spike and wave EEG activity that we associate with absence seizures. And clinically, these cats were having those types of events. And they were able to replicate these findings in humans too, that low frequency stimulation was able to evoke this, what is marked as here as LFS, this high amplitude, low frequency rhythmic discharges associated with clinical seizures. Conversely, if you deliver high frequency stimulation, which is again the sort of thing that we use in movement disorders as a kind of lesional network block, that actually resulted in broad desynchronization of the cortex. And that's the same type of stimulation that can result in reduction in seizures. And on the right here is one study that was doing this with temporarily implanted electrodes for seizure mapping. And so they had electrodes in a different nucleus of the thalamus called the anterior nucleus of the thalamus. And they found a similar phenomenon where they saw broad cortical desynchronization with acute stimulation of the thalamus. And this is starting to make maybe a little bit more sense that we are changing broad cortical connectivity patterns with this stimulation, not just defibrillating or blocking the signal. And a theme that I'll keep returning to here is, what conceptually does synchrony mean in epilepsy? I really think of epilepsy as a disorder of hypersynchrony. It's, much more complicated than that, but ultimately the patterns I think that we're looking for is decreasing this pathological level of synchrony or tendency towards synchrony in epilepsy. Okay, so that's acute stimulation. What is the effect of chronic stimulation on functional connectivity? So I'll give you two examples here. One is from the RNS device, one is from the DBS device, not specifically from the thalamus here. But the image on the left is a schematic from a study out of UCSF looking at a responsive neurostimulator device with electrodes placed in the hippocampus on the left and the right side, and looked over an extended period of time at connectivity within and between the contacts of these electrodes to see how is this network changing over time and how is it different in people who are having a good response to the therapy versus a bad response to the therapy, meaning people who are seeing a reduction in seizures versus not seeing a reduction in seizures. And so they looked at, this group was looking, I believe, at imaginary coherence, but you can look at a bunch of different connectivity measurements. And so they were looking at different frequency bands. I've shown here, you hear the theta frequency band, which the theta rhythm is a very prominent rhythm in the hippocampus. And what they found is that you saw a discrepancy between changes in connectivity between what we call super responders, people who had more than 90% reduction in their seizure frequencies, versus poor non-responders who see less than 50% reduction. And those poor responders were actually seeing an increase in the connectivity between the hippocampi versus the non-responders were seeing no change to a decrease in their connectivity. Again, kind of that idea of that very crude idea of more connectivity might be bad, especially between two areas that are kind of hotspots in someone's seizure network. The figure on the right looking at deep brain stimulations is actually a weird case of someone who had both a DBS and an RNS system but looked at differences in connectivity over time and with the deep brain stimulation system on and with the deep brain stimulation system off. And was able to show a little bit of a complex dynamic where stimulation of the anterior nucleus of the thalamus was driving decreases in connectivity within areas of the temporal lobe that are connected to that part of the thalamus, but not directly part of the thalamus. So kind of the picture that we're building is that what we're really doing with these types of brain stimulation, independent of whether it's DBS or RNS, is that we are changing network connectivity, network structures. If you're network neuroscience, kind of changing the strengths of the edges between these different brain nodes. All right, so let's talk about networks. So I think a little bit of context for this is one of the things that we do in epilepsy is to map the brain in order to figure out where seizures come from, and that can be useful for honestly the majority of patients that we see now who don't have like a brain tumor or a very clear structural problem for us to cut out. They just have a normal brain MRI and they have some EEG data that might be helpful, but we have to answer the question of where do the seizures come from and what would be the right surgical intervention for them. So, a common thing that we do is recording EEG from inside the skull, intracranial EEG. The way that we do that typically is with what's called stereo EEG, which are depth electrodes that are put into the brain temporarily to map specific areas of interest to understand where seizures are coming from. And so I'll call that stereo EEG or SEEG, and a lot of the data that we're going to talk about in this section is related to that. So this has been used for a long time. This has been actually used since the 40s. to map seizure onset. But something that people have been doing more and more recently is trying to incorporate thalamic in targets, not because we think that seizures come from the thalamus, but to understand how the thalamus is playing into these seizures and how it could affect our surgical decisions. The earlier thought about that was if the thalamus is a really strong part of someone's seizure network, if you were to cut out their temporal lobe, but their thalamus is still a bad player and is really still really connected to pathologic brain regions, they're less likely to be seizure free, and that's held up in large studies. But also, now that we have devices that we can use to stimulate the thalamus, the question is too, if the thalamus is really a big player in someone's epilepsy, could stimulation there be good? Now, now we come to kind of the cutting edge of this, which is a relatively crude cutting edge and gets back to the idea that there's a lot of low-hanging fruit here, which is that early seizure involvement is good for stimulation. If the thalamus becomes involved in a seizure shortly after seizure onset, at the site of seizure onset, that means that there's a big connection between the thalamus and that area of seizure onset, and that you should be stimulating that area of the thalamus to give the patient clinical benefit. And this is interpreted clinically as kind of, you look at these, what I call, think of those like kind of like swigglograms, is you're trying to see, how early do you see rhythmic activity in the thalamus? And this has really just been visual analysis for the most part. So there's a lot of computational analysis to be done here, and a lot that we've learned in the short time that we've been doing computational analysis. Kind of the most maybe a straightforward example of that is some of the work that I did with a colleague, Aaron Geller at Colorado, looking at ictal synchronization and how do we see that visually versus how do we see that across connectivity metrics. One connectivity metric that I brought up earlier was imaginary coherence, which is a way to look at phase coherence across phase connectivity across brain areas, and then the imaginary part is designed to eliminate zero phase signal. And then a secondary measure, which is Granger connectivity, which if you haven't talked about it already in the course, this has to do with directional connectivity and how are signals being communicated from cortical regions to thalamus and how are signals being communicated from the thalamus to cortex. Uh, so this has been explored by a few folks, and people have talked about, yes, if you see visually see changes in the thalamus, you see high amplitude spiking, or you see very fast activity, that this is accompanied by changes in connectivity. But what we found is actually visual analysis really fails to tell the story, that you often see changes in connectivity around seizure onset, even without seeing visual changes in the EEG. And so, what I'm showing you two pictures here. On the left side is this kind of flat map of the brain looking at electrodes on the left and the right side of the brain. And so these are these SEEG electrodes and these little boxes along here are those contacts. And one of these electrode contacts is in CM, the central median nucleus. And so we were looking at connectivity between the seizure onset zone and the thalamus here. And then we have these different levels, seizure onset zone and non-seizure onset zone cortical channels. And then we look at these measures, imaginary coherence, Granger connectivity from the thalamus to the cortex or from the cortex to the thalamus. And then we've centered this around seizure onset, which is in the middle of this panel. And then the y-axis here is frequency. Again, coherence is a value that is measured across differently across frequencies. And you can see this is an example of someone who didn't have very clear visual seizure onset in the thalamus, but had very clear increases in connectivity, both in imaginary coherence, but even more visible in terms of Granger connectivity. And you can see that in both directions from cortex to thalamus and thalamus and cortex, you had increases in connectivity after seizure onset. And we are really getting into some of the more weeds of this, but this turns out to be a relatively complicated thing that probably has to do with specific nuclei of the thalamus and the different relay patterns and the different phases of seizures. And then this is maybe a nice segue to talk about measures of connectivity that are not just seizures. So I guess my soapbox point here, which is going to motivate a lot of the rest of this talk, is that seizures are a rare state in epilepsy. So the brain is not seizing like 99.9% of the time in people with epilepsy. And so the brain networks are still abnormal. And so there are different sorts of measures of connectivity that we can glean, not just seizures. So this is a very nice work that was done by a group at Pittsburgh that we collaborate with, looking at a bunch of different measures of connectivity. And this sort of spans the functional connectivity realm, but also structural connectivity. So in the top left, this is structural connectivity. Structural connectivity is physical connections between brain areas, white matter paths, white matter fibers. Uh, Clinically, we can describe this using diffusion tensor imaging, and that's its whole own technical bin, but it allows you to quantify connectivity between different brain areas just from a structural standpoint. Uh, One way to look at this is this ictal synchronization, and so this bottom left figure kind of does something similar to what I was showing on the last slide, looking at connectivity between cortex and thalamus. But this is looking at a different metric. This is the H2 or H squared correlation coefficient, which is a nonlinear measure of synchrony between different brain areas. And this is describing an increase in synchrony in what they refer to as hodologically matched nuclei. In this case, the idea being that different nuclei of the thalamus are more connected to different brain regions. So they somewhat consistent color coding here. So this is the pulvinar here in blue. Pulvinar is very connected to posterior parts of the brain, parietal lobe, occipital lobe, temporal lobe, depending on what part of the pulvinar you are in. And so this, for example, was a patient with a parietal lobe seizure, and what they saw when they recorded from the pulvinar, and then the tan one is the anterior nucleus of the thalamus, is that they saw a big increase in synchrony between those areas, between the cortex and the pulvinar, but not between the parietal lobe and the anterior nucleus of the thalamus, which is really not well connected to the parietal lobe. Uh, they did something similar with stimulation. So there are different ways to measure resting state connectivity with electrical brain recordings, including stereo EEG. And so this uses evoked potentials. The idea of evoked potential is that you deliver stimulation in one area of the brain, or else outside the brain for that matter, and record the response in the brain. And so these specific evoked potentials are stimulation of the particular thalamic nucleus. For example, here they're delivering a particular type of stimulation to the anterior nucleus of the thalamus and then recording in different brain areas. And you can look at the magnitude of that response to characterize connectivity. And so they describe something similar, is that if you stimulate a part of the thalamus that is well connected to a specific brain region, that you see large evoked potentials. And then if you stimulate, for example, the anterior nucleus of the thalamus and you're recording the occipital lobe, which isn't really connected to it, you see minimal or no evoked potential, very low amplitude recordings there. And then the bottom right is another stimulation method, but more clinically relevant. So one of the things that we see in epilepsy is we see interictal spikes, so spontaneous discharges in the local field potential. And so these occur at some regularity. And so they describe the ability to do therapeutic stimulation and actually reduce the frequency of spikes, but again, only in what they call hodologically matched parts of the brain. So areas that where the connectivity between that part of the thalamus and the brain matched. So again, this continues to build this idea of where you stimulate is important, the nature of someone's seizure network is important, and so certain areas are probably going to be beneficial, certain areas aren't going to be beneficial. Probably connectivity to the seizure network is going to affect your ability to reshape the seizure network. So we're building into this a little bit. This is work that I've been doing with my postdoc, Mahmoud Alipour, who has been working with me to understand the relationship between connectivity during seizures and between seizures, so ictal versus interictal connectivity. And so, again, a lot of the focus has been on how early a seizure spreads to the thalamus. And so we have used this metric to look at early spread versus late spread versus no spread of seizures to the thalamus and describe this as latency between cortex and thalamus. And then we use this same H squared correlation coefficient, which was developed in the 90s and has been applied a lot to stereo EEG data. And this is very much in the early works. We've been operating on this hypothesis that probably the higher your connectivity is between the seizure onset zone and your particular area of the thalamus that you're recording from, probably you're going to see seizures earlier versus if you have poor connectivity, you probably see seizures later or not at all. We're very early in this process, and so you can see that relationship is not necessarily borne out by the data. But one of the things we've been talking about recently too is that this is probably a more complicated problem related to the structure of the anatomic connections here. So people talk about the pulvinar as being a more complicated nucleus with polysynaptic connections versus some of these like motor and sensory thalamus are more monosynaptic connections. And so really part of the state of the art of this literature is trying to figure out how much we can lump or split different thalamic nuclei. So, big ideas here to talk about thalamocortical networks is that effective stimulation probably depends on the stimulation target being a part of the seizure network. And that conversely, stimulation of that part of the thalamus will change the functional connectivity of the seizure network to result in clinical benefit. Right. Next, I'm going to talk about seizure cycles and states in epilepsy. So networks is sort of, we think of it as a structural concept, and then cycles and states is more of a temporal concept. My goal here is to talk about this in an area that we understand better, cortical onset epilepsy, seizures that come from the temporal lobe, which are, again, a big part of epilepsy in adults. And then after that, I'll kind of bring these ideas into thalamic stimulation, which is kind of a black box within a black box in epilepsy. So let's talk about seizure cycles. So people have understood for a long time that many people's seizures occur in cycles. This is, for example, noted in some women who have seizures associated with their menstrual cycle, and so that's a particular type of epilepsy called catamenial epilepsy. And so people have monthly seizures, but that's actually really quite common for people to have seizures on a monthly basis, or even people will say that they have seizures at a certain cycle of the moon, or that they will have cycles that occur with seizures more frequent at certain times of year. Even more common than that is that people will say that they have seizures at certain types of day. A patient that I saw recently just has seizures when she is waking up or just before she wakes up. So the presence of seizures, the presence of cycles in people's seizure frequency is actually quite common. And as we developed more and more longitudinal databases of brain recordings in people with epilepsy, we found that almost everyone has some sort of seizure cycle visible. This is helpful because one of the therapeutic challenges in epilepsy is that seizures seem very random. They seem to come out of nowhere. And so, of the people who don't notice that they have cyclic seizures, but do in fact have some sort of cyclic variation in their seizure risk, that could be useful, one, for informing people, and two, for developing devices that could potentially get people out of these higher risk states. So how do we think about this? How do we characterize this? So seizures, even if seizures occur somewhat randomly, we have the ability to record interictal events. So things like spikes in the local field potential or repetitive rhythmic discharges that don't necessarily manifest clinically. So this responsive neurostimulator device has the ability to track both those events as well as seizures, which it sort of automatically detects as prolonged abnormal discharges. Uh, and so, whereas someone might have these prolonged events, what they call long episodes, a few times a day, or a few times a week, or rarely every month, they are still probably having tens to hundreds of these interictal discharges a day. And so that allows you to reconstruct these cycles. I won't really get into the weeds too much of the signal processing aspect of this, but you can imagine that you have a signal that is a continuous signal that has multiple cycles that are built into it. Again, many people have daily cycles, but some people may have a cycle that's a week long. Some people may have a cycle that's a month long. Many people have multiple cycles built into this. And so this is a type of wavelet deconstruction that's used to identify particular cycles that are present within this interictal rhythm and then try to understand is there phase locking between seizures, so these long events, and this interictal cycle. So this is work that was really pioneered at UCSF in the late 20 teens to demonstrate the prevalence of this and really demonstrate the ability to track these. Now, it's not just that the seizures are linked to the cycles, but that there's specific phase locking, right? So if we think about a cycle as having four phases, so you have a part where your sinusoid is rising, you have a part where your sinusoid is at its peak, your sinusoid is falling, and then it's at its trough. The seizures really are mostly associated with the rising and peak parts of this parts of the cycle. And that kind of makes sense, right? That if your interictal activity, if your interictal spikes are becoming more and more frequent, that's a sign that you are shifting from kind of a good brain state to a bad brain state. And so that is when seizures across people tend to occur most frequently. Whereas we think about the transition away from the peak, so the falling phase and the trough phase are lower risk states. And so now we can start moving away from thinking of seizures as random, difficult to predict events, and we can start talking about a biological fluctuation of biological states. Um, uh, Put a pin in that, I'll come back to that. But when we talk about benefit of stimulation, we talk about effects of the cycles and stimulation, it actually seems to be quite important for when stimulation is delivered. So returning to this idea of RNS as a defibrillator device, if RNS is a defibrillator device, you'd expect that effective stimulation is given during seizures and then it stops seizures and then you're great. However, what has been found by some of our collaborators at Brigham and Women's is that when stimulation is preferentially delivered at those high risk states, like the peak phase of this, it's actually the least likely that people have clinical benefit versus if you were delivering stimulation during that transitional period where you're going from a low risk to a high risk state. And we're talking about a transitional period that's lasting days to weeks, that people who receive stimulation during that transitional period are most likely to be benefiting from seizure and most likely to have outstanding benefit from stimulation. And then more particulars, the effects of different stimulation parameters seem to differ across risk states. So, for example, low frequency stimulation seems to be better during high risk states versus high frequency stimulation seems to be better during low risk states. And so this is painting even more of a picture of maybe our devices should be designed differently. Maybe the type of stimulation that we should be delivering should be delivered according to the phase that we're in and are we delivering stimulation? How much stimulation are we delivering? What type of stimulation are we delivering? So that's effects of cycles on stimulation, but then let's flip the story to see, what are the effects of stimulation on cycles? We already said that stimulation seems to change network structure. Can we also change the temporal structure of these seizure cycles? So, the thing that's been reported by the Mayo Group is that stimulation of the anterior thalamus seems to be able to affect both the cycle period, so how long are cycles? Are they a week long? Are they 2 weeks long, 3 weeks long? As well as the amplitude of these seizure cycles. This is a relatively small study, but I'll show it to you in the top left. This is looking at a number of humans as well as a number of dogs with epilepsy who were implanted with clinical devices. And so they were able to show here fairly variable changes where there were differences between high frequency stimulation and low frequency stimulation on these peaks. And these peaks are what we're looking at as we're looking at a periodogram where we have period length on the x-axis and then amplitude of those cycles on the y-axis. Um, the more robust study, actually, this was a really nice study that was done by led by the company that makes the RNS device because they have access to a huge amount of data across a lot of sites as well as their pivotal trial, is to look at the effects of different medication trials on these interictal cycles. You can imagine these patients aren't off of medication, they are on medication, and some of them are doing well and some of them are not doing well. And during the course of this, they may be having introduction of new medications to see if they could be beneficial. And there's really quite a marked difference between a beneficial medication and not a beneficial medication. And so on the right, we're looking at these plots of cycle. These are counts of interictal discharges over time. This is time in months since the seizure medication was introduced, and the seizure medication was introduced at this yellow line, and you see some people just have a flatlining of their cycles, and some people have just a continuation, and some people have kind of a dampening of their cycles, and that relationship of flattening versus dampening versus continuation is associated with the clinical outcome in those. These are examples, but this was well demonstrated to be consistent across different medications and different patients. All right, this is the idea I wanted to return to about understanding seizures and seizure risk is not you are seizing or you are not seizing, but trying to understand this as biological differences of brain states. So brain, interictal discharges are again still kind of semi-random, spontaneous discharges, and so this was an effort by a group at Penn to describe more physiologic features of these different states, and so. These are some of the things that you probably talked about in your course, things about connectivity. This is a different measure of connectivity that they're using in this paper. I don't call it exactly which, but connectivity across different canonical frequency bands, theta, alpha, beta, gamma. And then to look at as well just other features like power within frequency bands. So if you take a Fourier transform and you look at the power within the alpha band or the theta band or the beta band, how do those differ between high risk and low risk states? And then, more conventional things like spike rate, interictal discharges here on the right side. That's what we've been looking at as a way to construct these seizure cycles. And they found that these physiologic features are actually quite different across these different states. That connectivity tends to be higher in these higher risk states and lower in the lower risk states. The power is higher. And then obviously that spike risk is higher. And so they were actually able to take this conglomeration of physiologic features and predict seizure risk better than just what we've been looking at in the past, just by looking at these interictal discharges. And then a key part of this too is that when they delivered stimulation, they acutely disrupted the relationship between the cycle phase, so rising, peak, trough, that sort of thing, and the feature, so connectivity or band power. And so stimulation disrupts this type of abnormal brain pattern. And again, I'm trying to build this story of seizures aren't occurring in a vacuum. Seizures are occurring relative to this sort of physiologic baseline, which we talk about clinically as seizure threshold. Seizure threshold, I think, for a long time was kind of this theoretical idea. But the seizure threshold, I think, is a real thing. It's a transition from a low risk state to a high risk state that is described by physiological activity of neurons. Looking at this in a different way, so this is some work that I did again in Colorado with Leslie Kay, is one of the neurologists there, kind of extending some of that work that was done at Penn to look at something called aperiodic activity. Now, aperiodic activity is activity that is related to the 1 over f slope of the power frequency curve. So this may be useful to explain with this image in the top right. So if you've seen, Fourier transforms of neural data, or honestly a lot of non-neural data, you can see that there's an inverse relationship between power spectral density, and frequency. And many people have just said this is sort of a physiologic property of the system, or this is just noise within the mathematical property of the system, or noise within the system that you see more power at lower frequencies. But This has also been related to neuronal level processes, so particularly the relationship between synaptic excitation and inhibition. And so part of that modeling work that's been done has suggested that that slope, the steepness of the relationship between power and frequency increases as you have more excitatory activity. And so people have explored this in epilepsy. I think intuitively, epilepsy is an excitatory disorder that's marked in between seizures by increase in inhibition. So this makes sense as a thing to look at in epilepsy. So previous work has looked at seizures versus interictal periods and found that seizures are associated with this higher slope, a higher level of excitation. And so the way there's several different algorithms you can do, which can just be linear fitting of this relationship in log-log space or more convoluted transforms to remove periodic peaks, so what we call oscillatory peaks in the power spectrum. And so this is using one of those more complicated spectral parameterization methods to derive this exponent, which relates to this slope of this 1 over f slope. So what we found using these RNS recordings from, in this case, hippocampus, is that this aperiodic exponent was higher during seizures, as was previously described. It's higher during interictal discharges, so spontaneous discharges that occur without symptoms. But it's also higher during those higher risk phases. So the rising phase of the interictal cycle, which is the most associated with seizures, was higher than the trough, than the falling phase. And if you look at this figure on the bottom, so this is sort of I think of this as sort of if you think about cellular neuroscience as like, spike triggered activity, think of this as seizure triggered activity. So if you take a seizure at time zero and you look backwards, you say, okay, what was the aperiodic exponent doing before this? And so you have this plots of these red and blue. This is red is the context ipsilateral to seizure onset. Blue is context contralateral to seizure onset. And you see in the 6 or so hours before seizures, there's a really marked increase in aperiodic activity. People will talk about, being able to see more spikes or whatever in the hours or usually minutes before a seizure, but this is really, shifting the time scale of when can you have a better sense that a seizure is going to happen. And then kind of similar to the work from Penn, that the aperiodic exponent decreases acutely after stimulation. And so this really adds, I think, another biomarker of seizures to try to differentiate seizures based on a biological state rather than spontaneous discharges that occur quasi-randomly or stochastically. All right, so next section is I'm going to take many of those ideas that we've established using more conventional recordings from the seizure onset zone and try to use this for thalamic purposes. So let's talk about thalamus and state changes because I spent a lot of time talking about states. So the thalamus is actually a really important part of the brain when it comes to state changes. It's a huge part of the brain in terms of, for example, sleep, wake cycles, and consciousness. There's a big transition between thalamocortical connectivity and hippocampal cortical connectivity between sleep and wake states. A big target that we talk about clinically is the centromedian nucleus, which is that area, the brain area of the thalamus that's really connected to central brain regions, so posterior frontal lobe, parietal lobe. It's also very strongly connected to the reticular activating system, which is sort of your consciousness and awareness on-off mechanism. And because CM stimulation is both used for epilepsy as well as disorders of consciousness, it's a really interesting target for us from a clinical standpoint, but it also means that we have to better understand the relationship between centromedius nucleus and these different state changes from an electrophysiology standpoint. So, this is more looking at neuronal activity, as well as field potential activity to try to understand differences between different phases. So two different projects, one on the top, one on the bottom. This work on the top was done by Meg Ryan, who was a resident, now was a medical student, is now a neurosurgery resident at the University of Colorado, looking at recordings of from the centromedian nucleus, single cell recordings from CM during surgery. So this is during the surgery for electrode replacement. We're actually able to record single cell activity. And these are different measures. NRMS is root means normalized root mean squared activity. So this is looking at kind of the background, the degree of background between the centromedian nucleus and other thalamic nuclei. And then looking at the frequency of spiking. Now, spike here means recorded single cell spike, like action potential type of discharge. And how do these differ between the centromedian nucleus and the not centromedian nucleus? But also, how do they differ between cases in which the surgery was done asleep versus the patient was actually had surgery and the patient was awake during surgery? And kind of unsurprisingly, based on what we know about the centromedian nucleus, is that neuronal activity was actually very markedly different between the wake state and the sleep state, because the centromedian nucleus is much, much more active during the wake state. We can see that with chronically implanted devices using this responsive neurostimulator setup. Again, we have these spectrograms where we have spectral power versus frequency, and we can characterize power spectra between states of wakefulness and states of sleep, or between day and night. And so you can see night being here, night being blue here, or that, similarly stage 1, 2 sleep being a somewhat analogous curve here versus eyes open or being awake during the day, that there is a shift in spectral power towards lower frequency during sleep, which is generally described in the EEG, but you can also see features that are somewhat expected, which is that eye closure, for example, is known to produce an increase in alpha power across the scalp EEG, and this pattern is replicated too in thalamic recordings. And so this, I think, really emphasizes the importance of physiologic state changes in understanding power structure. But it also becomes important when you're sort of on the spectrum between physiologic changes and pathologic changes, and how do you respond to biomarker changes to develop a biomarker-based device that's going to take one of these parameters or multiple of these parameters and decide what kind of stimulation you're getting. Another part of this too that I won't dwell on, though, is that centromedian nucleus and many of these thalamic nuclei aren't all one thing. Centromedian nucleus has a lateral part and a medial part, and they seem to behave very different physiologically, and they have different targeting properties. And the lateral CM behaves more like the motor thalamus that's next to it, and the medial CM behaves more like the parafascicular or interlaminar thalamus that's next to it. And the more lateral part has been described as a better stimulation target for epilepsy and has more connectivity with many of these cortical areas that we think of as the areas where seizures come from, versus the medial part has more connectivity with the basal ganglia areas. And so a lot of the work is related to kind of, how do we take this information together and figure out where we should be stimulating, but also, where should we be recording from? And are those the same thing? So we're trying to work towards what I think of as a biologically inspired idea of closed loop stimulation. Closed loop stimulation, something we've been talking about, is the idea that you're recording brain activity and that you're delivering stimulation accordingly. And this has been really worked on quite heavily in the Parkinson's disease space a lot in the last 20 years, to try to figure out how can we best deliver stimulation to target instantaneous symptoms and instantaneous brain state. So a problem here that was identified by Cameron McNair as one of the leaders in this field is that we assume that the biological signal that we're recording is the same as the place where we should be stimulating. So if we're recording some sort of pathological oscillation in Parkinson's disease in the basal ganglia, we're assuming that if that oscillation is pathologic, then we should stimulate there to make that pathological oscillation go away, which is, I think, a reasonable assumption. But, is this true in epilepsy? Does this even make sense? So this is kind of what we've been talking about too, but this is looking just at the recordings from this device. So these are raw voltage versus time recordings and different nuclei. So this particular system has 4 channels here within the centromedian nucleus, within lateral thalamus, on the right side within the central nucleus and within the lateral thalamus. And looking at these sort of discharges, so again, this device has a event detector, and this thing that I've marked out in red is the event that was detected where you see a transition from these to these high amplitude oscillations. These detectors, they can't be that computationally complicated because you have basically a computer that is implanted within someone, and so you can't be using a lot of this computational power all the time. Otherwise, you're going to have to be replacing its battery every few weeks. And if replacing an implanted device's battery is a surgery, then you don't want to be doing surgery on someone every few weeks. So you have kind of a computational cap here, just from a device development standpoint. And a couple of the uh, algorithms that are relatively computationally cheap that people have used are called line length, which is basically the amount of travel per unit time that your EEG baseline is doing. And so if it's traveling big amplitudes, that's a lot of travel distance. Or if it's going very fast, it's also traveling a lot of distance and you have high line length. Or different detector, which we call bandpass, which is basically a measure of rhythmic spiking activity, which generates a lot of spectral power within certain power bins, certain frequency bins. Now, if we use these detectors quite commonly and we have some idea of where the best place to stimulate is, where should we be detecting? Now, when people looked at the early trials of these devices, they discovered that even amongst the devices they were using to stimulate CM, a lot of the stimulation that they were delivering was outside of CM, or sometimes they were using detections that were outside of CM and delivering stimulation within CM. And we talked about how the amplitude of these of this cellular activity in these different areas is different. The motor thalamus is a much more active area than the central median nucleus, which is relatively paucicellular. So, like for example, should we be recording seizures in motor thalamus and then stimulating the central median nucleus? This was really not well understood. And so what we did is we mapped out the areas where there was most change in these metrics across anatomic regions to try to see if it corresponded with our ideal stimulation map or didn't. And so we're looking here at MRI images with the nuclei. So VL and VPL are part of VL is motor thalamus, VPL is sensory thalamus, medial thalamus, which is medial dorsal thalamus, as well as parafascicular, and then pulvinar, which is part of that association area. And then this is looking specifically at the central median nucleus. And what we see actually is that the area that we described in a previous slide, I'll show you back here, this area that's red associated with favorable stimulation outcomes is also quite close to the area where these detections are most prominent. And this actually, this is a nice fact as for us from a clinical standpoint and a device design standpoint is that the area that we're stimulating seems to be the area where we should also be detecting abnormal activity. And so we can sort of clinically program these devices accordingly. And then moving into so that's again that's a little bit more spatial ideas. Let's talk about cycles and cycles within the thalamus. And this is some work that Brian Garcia, who's one of your classmates, has been working on with me recently, but trying to extrapolate some of that work that we've done in hippocampal recordings and trying to classify these cycles within thalamic nuclei. Now, there have been some papers, I showed you the Mayo one before, that show that, yeah, sure, there are cycles in thalamic nuclei, but I would like to understand, do those cycles work the same way that the ones that we've seen across hundreds of patients with hippocampal recordings? Are they different? And can the properties of these cycles be used to direct brain stimulation and be used to change brain networks? So this is some of our relatively recent data that we're looking at. Things on the left that you've seen before, which is this periodogram looking at cycle length, the period in days on the x-axis, and the amplitude of those cycles on the y-axis, and looking at different peaks in this. So you see a very prominent peak at 1 day, for example, in this patient, and that makes sense because again, most people have some sort of diurnal variation in their seizure frequency. But here too, you see variation in cycle length. This patient's cycle is about 2 weeks long. This is, they see, you also see superimposed cycles. You see a cycle peak around 30 days. And the same relationship to try to understand where are seizures preferentially occurring during these cycles. And so for this 14-day cycle that this patient exhibited, you see that the long episodes are generally clustered towards the rising phase of their cycle, similar to what we've seen in hippocampal recordings. But a trend that we're noticing is that it's a little bit more complicated. We spent a lot of time talking about the relationship between seizures and abnormal discharges in the area that the seizures come from, but now we're recording from a different brain area that's not an area that we think about generating seizures. And how can we use this um, kind of like brain-centric hub recording? So we're thinking about the thalamus as a hub rather than where the seizures come from. How can we use data from the hub to inform how we deliver stimulation? Some work that we did looking at the effects of chronic stimulation on other potential biomarkers that we could use for stimulation. So uh, this is work that we've done, but different groups have seen sort of similar relationships, is that changes in the power spectrum can also seem to correlate with clinical benefit. And so using kind of a Parkinson's disease model, some people have looked at anterior thalamic stimulation, and if we suppress peaks that you can see, in Parkinson's disease, the beta peak, the beta band peak is anti-kinetic activity. It's associated with slowness and rigidity and stiffness. So stimulation of that, suppression of that beta peak with stimulation seems to be a good thing. So they looked at saying if we suppress the peaks that we see in the anterior nucleus of the thalamus, is that good? And they seem to find some relationship, not necessarily with physiologic peaks, but a group in Minnesota has looked at suppression of slow gamma activity and found that that seems to correlate with benefit. This is an example actually of a patient who has, again, this peak that you see, this physiologic peak that you see with the centromedian nucleus in the alpha range, which was actually brought out during a longer seizure-free period that this patient had. And so when we looked at these sort of seizure-triggered averages of power within these particular frequency bands, like this patient is sort of like this theta to alpha range frequency band, you see, or I get alpha to beta frequency band, you see that this physiologic activity actually was suppressed prior to seizures and seemed to rebound after seizures when the patient goes into kind of this refractory period. So again, kind of what we're doing is we are accumulating these different biomarkers that could be used to characterize healthy and unhealthy states as brain states rather than just probabilistic event states. Alrighty, so that's it for the data I'd like to show you. The things that I'd like you to take home from this is that brain stimulation for epilepsies works well, but that there's huge opportunities for improvement, that we can really set our sights higher to have more people be responders and have more people be seizure-free rather than just being better. That existing ideas about how thalamic stimulation works are really tied to outdated and sort of simplistic ideas of how neuromodulation works, and that neuromodulation probably really works by changing the structure of seizure networks, by changing connectivity, by changing these cycles, but also that the cycles have an impact on how seizures, how well stimulation works. And ultimately, taking all of these, sort of translational ideas, more biologically founded ideas, is likely to make this work well from a clinical standpoint. I could take your questions in a second. I'd just like to thank a lot of folks that I work with. This is not all work that I've done by myself. And so thanks to my research group, Mahmoud Ryan, as well as Mohammed, who's a medical student that works with me, and Arjit's a medical student who's coming into my lab. Uh, Women's been a big part of my own personal education in this, especially as someone who comes into this as a clinician and not a PhD. And then I work with a big spectrum of people from the clinical space to the research space at U Chicago, as well as at other places, University of Colorado, where I did my fellowship, and colleagues at Mass General as well. And so this is a very much a non-exhaustive list of the people who've been big mentors to me. So, with that, I'd love to hear any questions that you guys have. I'd love to talk about the ideas here, either getting into the nitty-gritty of some of the computational aspects of this, or talking about how signal processing can be useful in medicine.
Well, first of all, David, thank you very much for this talk. It's very inspiring.